Hello everyone. Today we are going to see about antibodies. Antibody is a large protein and it constitutes white globulin produced by plasma cells. It is used by the immune system to identify and neutralize pathogens. The pathogens such as bacteria and virus. These antibodies are also called immunoglobulins. The antibody recognizes a unique molecule of the harmful agent called antigen through the variable region. Structure. All antibodies share a basic structure. These antibodies are heavy globular plasma proteins or glycoproteins. The attached glycans are critically important to the structure and functions of the antibody. Each antibody is heterodimer and with a molecular weight of 150 kd. This is the basic structure of the antibody. It has a light chain and a heavy chain, constant region and a variable region. Now let me see about immunoglobulin domains. This antibody is composed of two identical helipolypeptide chains and two identical light chains. These are bounded through interchain disulfide linkages. Each chain is composed of structural domains which is known as immunoglobulin domains. These domains contains 70 to 110 amino acids. Heavy chains. There are 5 types of heavy chains: alpha, gamma, delta, epsilon, mu. Each heavy chain has two regions, one constant region and another one is variable region. The alpha and gamma chain contains 450 amino acids, whereas mu and epsilon chain contains 550 amino acids. Light chains. There are two types of light chains, kappa and lambda. These all antibodies have one of the two kinds of light chains. A light chain has two successive domains. One is constant domain and the other one is variable domain. It contains 211 to 217 amino acids. This image represents the sites of immunoglobulin. Here you can see the light chain, heavy chain, disulfide bridges, antigen binding sites, variable region on heavy chains, constant region on light chains. Everything you can see in this image. Next is different classes of antibodies. There are 5 classes. They are immunoglobulin G, M, A, D, E. We will written as IgG, IgM, IgA, IgD, IgE. These antibody classes are named according to the heavy chain types. First we see about immunoglobulin G. It is made up of 80% of serum antibodies. It has a half-life of 7 to 23 days and this IgG is a monomer and it has 2 epitope binding sites. This is the only class of antibody that can cross the placenta and enter the fecal circulation. Functions of IgG. It gives immunity to newborn, it neutralization of toxins, and IgG3 binds to Fc receptor by the process of phagocytosis. Next one is IgM. It is made up of 13% of serum antibodies and it has a half-life of about 5 days and it is a pentamer and it has 10 epitope binding sites. Some of them are monomer. It is the first immunoglobulin class produced in a primary response to the antigen. Functions of immunoglobulin M. Activation of classical pathway. It defends against the multivalent antigens and it acts as a opsonin. Next one is immunoglobulin A. It is made up of 6% of serum antibodies and has a lifespan of approximately 5 days and it is a dimer and has a 4 epitope binding sites. They are found mainly in body secretions such as saliva, mucus, tears, milk, etc. Then the functions of immunoglobulin A are it is a secretory antibody and it effective against virus that causes influenza and there is a production to infant gut. Next one is immunoglobulin D. They are made up of approximately 0.2% of serum antibodies and it is a monomer and it has 2 epitope binding sites. This class antibodies are found on the surface of the B lymphocytes. Functions. B cell activation and it acts as a receptor for antigen binding. Next one, immunoglobulin E. It was discovered by Can T. Ishkasha, and it is a very low concentration in blood, and it contains a small percentage of lymphocytes. Functions of immunoglobulin E. It is responsible for the immediate hypersensitivity, and it binds to Fc receptor and basophils and mast cells, and it releases substances like histamine, vasoactive mediators. And these are the structures of antibodies IgG, IgE, IgD, IgM and IgA. Thank you friends